Perfect, thank you. Welcome everyone to Managing Problematic Diseases and Insects on Hemp. I am Brenda Palencia. I'm a marketing intern at Marone Bioinnovations. Before I get started, I would like to remind everyone that we do have CEU credits, but you must take a quiz at the end of the webinar. I will be providing you a link as well as a QR code for you to take that at the end. And certificates for that will be sent out within 1 week from today. And for Washington and Oregon, this is just a reminder that you must participate in the comment section halfway through the webinar by adding in the chat your name, license number, and accredited state. Lois will also be providing a quiz question that you will have to answer in the chat. We will bring that up later. If you have any questions, let us know. Before we get started today, I would just like to remind you all that Marone Bioinnovations is a publicly traded company, so we are required to remind you that any information discussed in this presentation should not be solely used to determine your interest and/or level of investment in its stock. We encourage you to conduct your own due diligence when making financial investment decisions about Marone Bioinnovations. Now we can introduce our speakers today. Today we have Lois Hawkins, who is the Western Cascades Territory Sales Manager at Marone Bioinnovations. Lois is an experienced technical support manager with a demonstrated history of working in the biotechnology and agricultural industries. She has over 10 years of experience in commercial agriculture product development and technical sales support, as well as 15 years in crop protection management. As a licensed Oregon PCA and Washington and Idaho demonstration protection management, a research and private applicator, she has extensive crop protection, production, and integrated pest management experience driving product demand through field development, education, and training. Lois is skilled in leading disruption implementation, field trials, integrated pest management, and precision agriculture. Lois is a strong support professional with a bachelor's degree focused in biological sciences from Southern Oregon University. Today, we also have Steve Bogash. Steve began his career as the owner and operator of Greener Horizons, a garden center, nursery, greenhouse, and landscaping operation in Maryland, before serving nearly 20 years as a horticulture educator and researcher from the Pennsylvania State University Cooperative Extension in State College, Pennsylvania. Since retiring from extension service, Steve joined Marone Bioinnovations as our Northeast Mid-Atlantic Product Development and Territory Business Manager. His territory runs from Southern Virginia to Maine to the western edge of Ohio. Steve oversees several dozen university and private research company product trials, as well as many on-farm demonstration trials for Marone Bioinnovations. Today we have a special guest, Dr. Govinda Shrestha. Govinda has been named Statewide Hemp Extension Specialist at Oregon State University's Global Hemp Innovation Center. Govinda holds a PhD in agrology and a master's degree in agrobiology from Aarhus University in Denmark. He has extensive agriculture experience in the US, Europe, and Nepal, where he grew up on a farm. He has most recently worked as a postdoctoral scholar in the Integrated Pest Management for Potatoes at the OSU Hermiston Agriculture Research and Extension Center. He was also a postdoctoral researcher at the Western Triangle Ag Research Center in Montana, where he specialized in pest management for wheat and alfalfa hay crop pests. Govinda is based at the OSU Southern Oregon Research and Extension Center in Central Point, where he provides statewide extension and outreach during the peak outdoor growing season. So thank you to all our guests for joining us today. For today's agenda, we will start by introducing a little bit about Marone Bioinnovations. Then we will pass it over to Dr. Govinda. We will then have Lois and Steve have a little bit of biofungicide introduction as well as data, as well as biological to control arthropod pests in hemp, along with data. We will then conclude with a Q&A as well as provide you with information for your CEU credits. For our company overview, we would like to remind you all and give you a little bit of information about Marrone Bio Innovations. It was founded in 2006, and we do have our headquarters in Raleigh, North Carolina. We do have a manufacturing location in Bangor, Michigan, as well as an R&D Center of Excellence in Davis, California. We do have nationwide sales and technical support, global distribution, and 500 patents worldwide. And this is our CEO, Kevin Helash. This is a quick overview of our portfolio. Starting with crop protection, we have fungicide, bactericide, disinfectants, ag and oxide. For fungicide plant health, we have Regalia and Stargus, which we will be covering today. There is also Haven, a crop stress reduction. And for insecticides, we have Grandivo, Venerate, Magistine, and Salto. For plant health, we have a product pace setter, and we have multiple seed treatment and foliar fertilizers as well. Now I will pass it over to Dr. Govinda.
Thank you, Brenda. Is that my volume and sound is okay, Brenda?
Yes, we can hear you okay.
So you're gonna move the slide, is that right?
Yes, just let me know when you'd like me to click.
Yeah, maybe you can click now, yeah. So first of all, I would like, and good morning, everyone. First of all, I would like to thank Marrone Bio Innovation, and especially Lois and Marina for providing the opportunity today to talk on a hemp pest complex in outdoor hemp. As Brenda mentioned that I am a hemp extension faculty with Oregon State University, and I'm located at Southern Oregon Research and Extension Center in Central Point, Oregon. And I'm also the faculty, also the associate faculty with Global Hemp Innovation Center. Can you move another slide? Brenda? Are you there, Brenda?
Yes. Would you like the next slide after the outline?
Yeah. Okay. Yeah. So this is okay. I will just tell you that so this slide represents the outline of my talk. No, go back.
Okay. Perfect.
Yeah. I will let you know. So this slide represents the outline of my talk. So first, I will briefly talk about Oregon State University Global Hemp Innovation Center for those of you who don't know about Global Hemp Innovation Center, and then I will focus on a hemp pests complex survey that we did in outdoor hemp field in 2021 in Oregon, and also I thought to share my field observations about the hemp pests that I or other faculty member working on hemp observed in outdoor hemp in 2021. So can you move to the next one? So for those of you who don't know about Global Hemp Innovation Center, this Global Hemp Innovation Center was launched by Oregon State University in 2019 by Oregon State University to help and support the newly formed hemp industry. And one of the goal or one of the promises of this the center is to advance the research of hemp and its market potential across the multiple diverse industry and research field to show up the growing international demand for the innovative approach to food, health, and fiber. Can you move another one? So, if you wanted to know more about the Global Hemp Innovation Center, please feel free to contact Jeff Steiner, who is Associate Director of the Global Hemp Innovation Center. Also, you can reach me as well if you want to know more about the Global Hemp Innovation Center or wanted to contact or wanted to connect with Jeff or other faculty working on a different different part of the hemp. And also I put like a link there, YouTube link, and also website, so where you find out more detailed information about Global Hemp Innovation Center. Now I would like to move to my real talk. Would you want to move to Brinda? Brinda, can you move on the slide?
Are you able to see Hemp Pest Complex Survey?
Yeah, yes, now I can see that. Now, this is now the second part of this is the main part of my talk. So I think it's giving you a little bit background about why it was necessary to do the hemp pest complex survey in Oregon, maybe also in other part of the United States. So so as we know that hemp is a relatively new crop, and there is little bit or no information about what kind of insect pests can be attracted to the hemp, or what kind of the insect pests have potential to cause the damage to the hemp. So that kind of that's why there is a need of like establish the baseline information that can eventually help to develop the hemp IPM program in Oregon and Pacific Northwest. So that was the kind of the goal of the whole study, is to establish the baseline information. And we did on 2021, and we will be doing this year and several, at least 2 3 years more, to understand the hemp pest complex in Oregon. And I believe that data would be relevant to the Pacific Northwest mainly. So now I would like to kind of go through about my data, that what I found out in 2021 field seasons, and also would like to kind of provide the takeaway from this data, what we think about it in that way. So can you move, Brenda? So a little bit about like about how we did the survey. We did the insect pest complex survey in a pretty simple method. So as you can see on a picture on the left, this is the alloy sticky card. We use the alloy sticky card to monitor the flying insects such as aphid, lygus bug, and thrips. Lygus bug, aphid. It's just a couple. Okay, aphid, lygus bug, and thrips. Okay, sorry, something happened. Okay, thrips and we use the beating seat, like you see in the picture on the middle, to monitor the particular insect pests that are colonizing really on the plants. And also we monitor the corn earworm. I think this is the one of the pests currently being considered as a major threat to the hemp industry. So we use the Heliotis pheromone trap to monitor this pest. Basically, what we did is that we monitor the insect pest complex from mid-July to mid-October or late September, and we did that surveying at a 1 weekly interval, and we have 3 to 4 fields that we monitor. Now I would like to share what kind of pest complex we found out using the different the monitoring tools. Can you move, Brenda, another one? So just a little bit. So moving to the results, like the picture on the left, the graph on the left is what we found out, what we find in a LSTP card, and picture on the graph on the right is what we found onto the plant using the beating seeds. On the horizontal line on the both graph represents the insect species or insect compositions. On the vertical line is the total average count per trap, meaning that how much total number of average that we count per trap at the end of the seasons. So from the alloy sticky trap, what we learn is that what we caught or what we learn is that thrips can be at thrips we capture the thrip at highest level, followed by aphid, leafhopper, and lygus spot. And the thrips number were about 2 to 40 times higher than other insect category or other insect types. But on the other hand, on a beating seeds, meaning that on a real plants, we found the aphid as the major dominant insect, followed by lygus bug, leafhopper, and thrips. And we also captured these false incy bug, flea beetles, and cucumber beetles, but they were at a low number. But they were at a uh, low number scales, meaning that very low number. So here, one thing what I wanted to point it out on this graph is that the aphid is the main insect that we we found out in our beadseed samples. And also one thing what I wanted to point it out is that we also found out both aphids, nymphs, and adults, not only the aphids but also the lycospores. So what it tells is that uh, this insect pest can reproduce on a hemp plants. For the leafhopper, we found out the few nymph of the leafhopper, but we really don't know exactly, like they are really success, they are really making success to reproduce the nymph or not. But just to give you an idea about the aphid and the lycospores, nymph and adults both were found in good numbers. So the takeaway is the aphid could be the main pest, and followed by lycospores. That's what is the takeaway of this slide. Can you move another one? Also, we closely monitor the leafhopper species in our survey. The reason behind is that there is a really, in this last 2 years, there is a really concern about that leafhopper have the potential to vector or transmit the disease. So we thought that we wanted to monitor very closely about the leafhopper species. The graph on the left is the leafhopper compositions that we found or that we capture on a sticky card. Graph on the right is leafhopper species that we found out on a real plant using the beating seeds. So there are the three leafhopper species that we mainly found as a major one. One on the yellow one is the beet leafhopper. That is currently the major threat for the hemp industry because it's vector the beet collector pathogen or disease. Another is the potato hopborn or potato leafhopper. That's a pretty green color. Another is Stratagalia. So these are the three common the leafhopper species that we recorded. And in the yellow sticky card, we found out the potato hopper as a major one, and other one beet leafhopper and Stratagalia were found at low number. But if you look at on the beating seed, meaning the real plants, the Stratagalia were the most the common one, followed by a pretty similar number of the beet leafhopper and the Impersca. Here, what I wanted to point it out is that even Stratagalia, this is the species that we found, but there is nothing, there is no report about this leafhopper can cause the damage or can vector the plant pathogens. So basically, this is good news for everyone, but we never know what will happen in the future. Another Impersca is also considered a minor. It can when you feed on the leaves, it can cause the kind of burning types of things. But there is really not much there is really few there is really in this pest this insect is really considered as a minor insect, so basically it's not considered as a huge pest. So and only the considering is the beet leafhopper. So that's a one that we currently been considered as a major insect pest on the hemp. Can you move another one? And also in our study, we also monitor very closely the cannabis aphids. I believe if you are a hemp grower, if you are a greenhouse hemp grower, you already knows about what the cannabis aphid. Cannabis aphid is a huge problem, particularly in greenhouse productions. And just a little bit about the background of this cannabis aphid. Cannabis aphid is originated or is from the Asia, and then it moved to it introduced in the US. So currently this insect pest is considered as an invasive pest species. To my knowledge, this insect is present in many states. As an example, in Oregon, Washington, and Colorado, Virginia, these are the common states that I know this insect is present. So we also monitor this insect very closely, since there is not much information about whether it first like present and what level it presents. So there is not much information outdoor condition. So the picture or the graph on the left is yellow sticky card data. So on the horizontal line is the cannabis aphid, that is the Phorodon cannabis and other aphids. On the y-axis, meaning the vertical line, is that the number of the total number of the cannabis aphid, total average number of cannabis aphid per trap. So what we found or what we expected, because we use yellow sticky trap, it can capture different types of aphids because the aphids are more attracted to the yellow sticky card. So we found out the use more than double number of the other aphids, but also we found the cannabis aphids. So meaning that it tells that we can capture the cannabis aphids with yellow sticky card, and cannabis aphid is present in Oregon. That's clear from our study. And can you click the slide? Yes. So, interesting, what we found out on a plants is that we found several aphids, but the cannabis aphid is almost like more than 4 times than other aphids. So basically, like cannabis aphid can be the major pest on a hemp, major pest or major insect that we found out. So when if I'm a grower, if I think about aphids, probably I will be encountering mainly with the cannabis aphids, and other aphid would be at a minor level. Can you move another slide? So also we are interesting to know exactly that in when and what time in the season this aphid arrive or reach the peak. So the picture graph or line graph on the left is just to kind of seeing that what line graph in the line graph, the horizontal line is the week that we monitor. On the vertical line is average count of aphids per week. So basically, in the LST card, what we found out is that aphid started to come up during the kind of early August or early to mid-August. That's the early flowering, and then they started to kind of started to get a peak, and they reach a peak late August or mid-September, and that is fitting that is coinciding with the flowering time or the heavy flowering time. That's what we found on LST card, and also we have pretty similar pattern on a plants, and also we started to see like a some few number on a early to mid August. Then they started to peak, and I wish that I could have taken the data from September 17 because of the time constraint. I couldn't take it, but I would like to mention here that one point is that we might see the huge number of cannabis aphid during the flowering time. So the question is that whether the cannabis aphid we are seeing in a plant are really causing a damage or not, because one of the things is that we are more concerned on the flower. So maybe that's a product that we want to harvest. So there should be like uh, some damage going on, but to my knowledge, I don't have any data that can tell. So the future study would be very interesting to know that how many aphids is kind of the considered threshold that tells about like, okay, these are the five if I get the 5 aphids per plant, or if I get a 10 aphids per plant, and that relate with the application of any kind of biological product, but we don't know at. And also another interesting thing is that why the cannabis aphid is more likely coming on a flowering time. Is there any correlation between the flowering, like stickiness, or what kind of relations going on? I think there are so many unknown factors, but it definitely tells from our study that in a last year, in a 2021, mainly the cannabis aphids are catching up at a peak level during the mid flowering time. So can you move another one? Yes. So moving to the cornearum, I think everybody knows when you work on the on a hemp is a cornearum is a serious insect pest. Just to give you a little bit more information, if you are not familiar with cornearum, cornearum is not a new pest for US because it's a native to North America, and it's known to the pest of several crops, but more importantly with sweet corn, cotton and tomato. And nobody thought about cornearum can also like the hemp, but unfortunately it turned out that cornearum also like to feed on hemp. So this is the graph. This is line graph that the data from our cornearum monitoring data that we did last year. In overall, we didn't found the huge number of cornearum. The cornearum number were pretty low. The number ranged from 1 to 4. But if you look at the graph carefully, there is sort of like a peak, that coming up almost on a mid during the flowering time. They are kind of we can cast the high number, so that's the time also the flowering. And also I'm thinking about other crop because in the corn they are more of also their number are more on the silk. So it makes sense that cornearum in general like the flower. So that could be the reason that why we are catching up or what there is a peak during the flowering time. So, but one of the takeaway is that we didn't find a lot of number of corneoram in the traps. So then comes to the question is, can you move to another one? Can you move? Okay. Then question is that whether it's a pest or not. Unfortunately, we found out huge number of caterpillar and corneoram feeding on a hemp. And we are kind of puzzled because we found very low number of corneoram moth on the traps, but the damage is going on. Lots of caterpillar are seeing on a crop. So the question is that because many crop there is a threshold level, and we don't know exactly what number can trigger the we can say okay, if we find out 2 corneoram moth per trap, that would the time that you want to apply the biological product. We don't know exactly. Maybe another question would be, is that a well enough that we can rely on a moth to monitor this pest? Maybe not, because maybe the moth monitoring tools, that the permanent monitoring tools only maybe tell you that, okay, they are going to active. Maybe you have to scout to the really on a plants that you can start see to the eggs or caterpillar. So there are certain like a certain uncertainty about how we can best bestly manage this pest. But definitely one of the takeaway is that I believe that the threshold would be low number, like because in other crop in a corn, like there is like a threshold might be different. So we need to determine that what number really tells us that on a trap that we can start to apply the biological product. So it's really not known. But what I'm hearing from the grower, if it's what I'm hearing from the grower is that if they started to see some number in a flowering time, they always prefer to spray. That's the grower perspective. But from the science perspective, we don't know exactly that how many number we can really consider at the time that we can start applying the product. Can you move another one? Can you? Okay, good. And just to share you the picture that I took last year, and on the picture on the left is a young caterpillar I started to see in the early flower, and it's like a mature caterpillar that I kind of catch and I think late August or mid September, something like that. But on the right picture is that like a real corneoram damage, as you can see that the bud rot. Just to give you an idea about feeding damage, really corneoram. What happened is the corneoram feeds on a flower. What happened is that then when it starts through that and it opened the rooms for entry for the many pathogen, and as a result that like our flower is going to rot. So normally, we are kind of making the environment fit for the maybe disease or other several aspects. So it's kind of causing an indirect way because you don't want to make it your flower bud to kind of rotting. So that's normally the major issue with this corneoram. Corneoram presents or corneal feedings turn into the flower on a bud rot. So that's one of the major issue with the grower. And I think you have to control the corneoram effectively if you don't want to see that kind of bud rot issue in your fields. Can you move another one? Now I would like to just to give my my field observation that I saw. And one of the issue, Lois might talk also as well because she's also associated with that problem. So there was a one corneoram. One of the grower had a the issue with corneoram. Actually, I visited at least 2 times or 3 times on his farm. In the first time, we didn't realize that he has any corneoram. He has any issue with the wireworms. Then when we started to dig the seedlings, we saw that the roots are packed with the wireworms. And then we figured out that why it's happening is that I think that the wireworm are feeding the roots, and that is making like a plant as like a nutrient deficiency because there is not a root growth, and then plants are looking as sick. And if you don't know about wireworm are soil-borne pests. They are known to feed on a variety of crop like wheat and potato. And one of the major issue with the wireworm is that when you have pasture or wheat rotation, that kind of in that area you can see the wireworm. So I just wanted to point it out is that wireworm can be a pest, but not over throughout the landscape, but maybe in some field you can have a like issue with the wireworm. So then I'm thinking that that pest also be considered as a major pest in hemp because what happened is that if you see the wireworm infestation, This plant will not die, but you will not get a good yield. Maybe number of flower per plant is less. So that, indirectly or directly impacting the, your crop quality, or no, impacting your crop yields. Can you move another one? I'm taking a long time. I just wanted to point it out quickly, like, there is a one pest called, I mean, I already talk about beet leaf hopper. Beet leaf hopper is a pest because it's vector, the beet colita pathogen. And this is a really challenge, in Oregon, and I believe in many other states. What happened is that, this transmits the virus called beet colita pathogen that makes the plant stunted and leaf stuffling. And what I observe personally is that when there is a really huge infestation of beet colita pathogen, the flower will be not formed there. They are sort of like kind of the ball type. I don't know how to explain it carefully, but looks like a ball, and then you don't see, like, you don't see any flower. So it's really impact that, the crop quality, I think, crop yield, and maybe also the quality as well. And also I want to let you know that, there is a statewide disease survey going on. I will talk on another slide by Cindy Oakham, who is associate professor and plant pathologist. So we are currently doing, like, the similar to the insect pest survey. We are also doing this disease survey, and at the end we'll have very good information about what are the major, the disease in a hampers. Can you move another one? Yes, just to give you another example that, that I took the picture. It's another is like a fusarium, particularly in mid in the season. I mean, if you are the hemp grower, you might already notice about these things. The plants looks completely healthy in early season, but when the flowering start, you can see the, the not a lot, but some plants are dying like that, and that's a fusarium. And I don't know exactly which species, but, there is a kind of one issue with fusarium that's also kind of put us on a on our mind, or we need to work on more on that kind of soil-borne pathogen on hemp. And also botrytis at the end of the season, you can also find out some issues depending on the weather, depending on the year, but definitely that also have a one, considering pest or disease on a hemp. And here I can you make it one more click? Yes, and also, like I mentioned, that I would like if you want to know more about disease things, I would definitely recommend to contact Cindy Oakham, who is a plant pathologist, who is leading the hemp pathology group with Oregon State University, and definitely we can give you the lots of information about what are the major disease, what are the minor disease, and what our study looks like across the Oregons. And and feel free to reach out me if you wanted to connect with her, or you can connect directly with Cindy Oakham. And with that, I would like to finish my talk, and please let me know if you have any comment, suggestions, or any feedback. Thank you, Brenda.
Thank you so much. We'll take a few minutes. If we have any questions, please type them in the chat for Dr. Govinda.
So
Brenda,
do you want go ahead?
I think we already had one question, and Cameron Blackford asked, "Did you find any mites, Govinda?"
No, unfortunately not. I mean, that's a good thing. We didn't find mites. That's a good, yeah. Last year, not. Also, one thing I would be very honest with you that sometimes mites are hard to detect. I might not have like clearly, like, spend too much time, but not like not much. But definitely, like spider mite and russet mite are kind of like, in our radar to monitor. But I was talking with from the US system, University of California, who did the similar survey, and they also didn't find a lot last year. So like, I mean, we didn't find. That's what I want to say. There are a couple of questions. Do you want me to reply, Lois, or what? The talk. There is one question. There is one question about, like, perhaps there is a one question from. I just wanted to read it from, like, what is? I'm just working on the chat box from the CB. Any product or spray to add the killing the cornearum or caterpillar, perhaps in early stage of development. I believe Lois and they will talk about the prospective. But if you look at on literature, they are telling about, like, the BT product, particularly Azua And also Venerate also, I think there are some data that coming off from the modern biome. They're also showing some sort of positive results to control. But I think there is a need of lots of more research on like what product we can really use to control. That's what I'm seeing is that. But definitely BT is the one that everybody think about it, as a major tools. And also there's a one question about trichogramma wasps for caterpillar eggs. And one of the things is trichogramma is that yes, trichogramma are known to be the good biological control agent for parasitic wasps that used for the cornea. But also what I wanted to point it out is the trichogramma is picky, parasitoids. Sometimes it depends on the crop. Like they might work on the corn, but they might not work on the ham. So we don't know exactly that whether the trichogramma wasps we can use as a parasitoid, as a biological control agent. But definitely, these trichogramma are known to be the use as a biological control agent for the cornea. Is there any more questions that I can take it here?
We have one more question from Robert. He asked, what further study is needed to establish treatment thresholds for pests you discussed today?
That's a fantastic question. Really, really, Robert, I think I like your question, this question, because I think first thing is like, if I think about like the cornea, is really we don't know exactly like how many number of moth that we can say that that's a threshold number. Basically, like we need to correlate the number that we study would be number of correlation of moth with respect to the caterpillar number into the plants. So that kind of study might help in that to determine that, okay, that these are the 4 cornea moths that we found. Maybe in that time you can see the damage, because one cornea female moth can lay the egg up to 3,000. Remember, we are a very small hemp grower. So maybe one moth would be enough. We don't know exactly. Like I was working with other insect, like back in Montana, called orange with blossom leaves. If you get 1 or 2, that's enough. Per plant, 1 is enough. You have to spray just 1 or 2. So it depends. So I don't have any right answer, but definitely there's sort of like a correlation with the damage of the number of caterpillar on the plants and number of moth on the pheromone traps. So that's kind of studies really really needed to really tell. And because this pest is coming up at the time of flowering, and you don't want to lose your flower, that's the one like you wanted to harvest. These are tricky insects. That's the one thing I was thinking in my mind. And also I was talking cannabis aphid. I think that also, like, we don't know exactly how many number of aphids would relate to the damage. So I think, and that aphid also comes, that I talked in that my slide, that they also come in the flowering time. We need to know the number. So basically we need to determine sort of like how many number of aphid present on a plant, and how many number of moth on a trap can really cause the damage. So that kind of study is required. But definitely, yes, that will help us at some point in the future. That's my thought or takeaway on that kind of threshold study. Is there any more, Brenda?
That looks like it for now. For everyone attending today, Dr. Shrestha would really appreciate if you took a survey that's in the chat section. There is a link. If you could take that, that would be great. If we have any more questions, we will send them to you. But thank you so much for joining us today.
Great, thank you. And I will be here and I will be watching the talk. So feel free to kind of chat me or anything you have a question to ask me. And I have my cell phone number in the slide. You can email me or call me. And I will appreciate that if you kind of fill up that survey form. That helps me also in my career. So please feel free, like 1-minute survey, a quick survey. Yes. So I'm now mute my bottom, so you can go ahead with your further work.
Yes, yes, please. Thank you. All righty, I will pass it over to Lois and Steve.
Thank you, Brenda. Thank you, Govinda. Really appreciate all the good information. So we're about halfway through the webinar. And so before we go on, I need to do the quiz question for those of you who want Oregon or Washington credit. So first of all, be sure if you're signed on anonymously, I need you to put your name in the chat box as well as the information for the response. But I need your name, I need your license number and the accrediting state, so that I'm the one who actually submits all those credits for Oregon and Washington. It really helps me if you tell me which state it's for. So your question is, what was the most detected hemp pest in Dr. Govinda's pest survey? So I'm gonna go ahead and let you, those of you who are on from Oregon and Washington, work on that in the chat box. If you have any problems accessing the chat box, use the reaction box next to it to raise your hand. But otherwise, I'm gonna go ahead and go on because in the interest of time. So Brenda, next slide, please. So today we're going to introduce some products that are EPA registered or have some state approvals for use in hemp production. We have 5 main products to discuss: Regalia and Stargus Biofungicides, JetAg, which is our Peroxy acetic acid disinfectant, and Grandivo and Venerate, which are 2 bioinsecticides. As you can see, the regulatory status of these products does vary by state. So in the interest of time, I'm not going to go into details here, but I just want to remind everyone that be sure and check with your state to find out whether our products, or actually any other product, is registered on hemp before spending the money on them. So next slide, please. So to alleviate some confusion when you're looking at Marrone Bio's product labels, on some products you're going to see an ag label, and on others you're going to see the CG initials on the end of the product, and I want to explain the differences. So CG actually stands for Cultivated Garden, which is our line of products marketed to the greenhouse and cannabis sectors. These products include the same active ingredients as their corresponding ag labels, but they do go through an extra level of testing to verify that there are no contaminants that could contribute to a hemp or cannabis pre-harvest testing failure. Also, CG products are available in different container sizes, and that's to help fit the needs of the small farmer. Brenda's included here a snapshot of our small tank mix use guides, and this is for that smaller grower or home grower that does need to adjust the labeled rates, which on a lot of the labels are as per acre, and you need to adjust those down to per gallon. So that is available on our website, and you can go in anytime and grab that small tank mix guide for your use. Next slide, please. So let's take a look at the Marrone products that can be good additions to the hemp growers' arsenal against the pests that Govinda talked about. We're going to start by briefly introducing the biofungicides. Stargus is our live bacillus. It's an Amylolocophaceans strain F727 product, which is unique. And as with many biologicals, it is best used as a preventative or early intervention fungicide. It is broad spectrum and can prevent many of the diseases that can attack hemp, including some of the most common ones like gray mold, fusarium, hemp canker, hemp leaf spot, southern blight, and verticillium wilt. It has multiple modes of action, and I'll present that information in the next slide. And as with all of our products discussed today, because I'm not going to mention this again, it is OMRI listed, has zero pre-harvest interval, has 4-hour REI interval, and requires minimal PPE due to its safety profile. So these are great products for using right before harvest or when you need to get back into the field in short notice. Next slide, please. So Stargus has 3 modes of action, which is really important when you're discussing efficacy and resistance management. The first, which is doing a lot of the heavy lifting, are the active molecules, especially the peptides produced during fermentation that attack spore germination, somewhat like a contact kill. Also, you get leaf and or root colonization of the living bacillus spores, which forms a protective shield against the pathogens. And the third mode of action is induced systemic resistance, which helps the plant defend itself against pathogens. Next slide, please. Next, I want to introduce Regalia Biofungicide. And in contrast to Stargus, which is a live bacillus and its fermentation media, Regalia is a plant extract and its fermentation media. So its active ingredient is Reinutria saccharinensis, and it's commonly known as giant knotweed. The fermentation products in the formulation provide broad spectrum disease prevention. And again, it's not killing anything, so this is truly a prevention product. It can be tank mixed with many other products and has a wide range of applications to fit the crop stage and or disease specific timings. Next slide, please. Regalia, like Stargus, has multiple modes of action, including induced systemic resistance and systemic acquired resistance. And that signals to the plant to produce several antimicrobial compounds that enable treated plants to prevent pathogen growth. Induced systemic resistance also signals to the plant to accumulate more lignin, and that strengthens the cell walls against penetration by the pathogens. An additional benefit is the enhancement of the target plant health. So what you spray it on, it increases structural integrity and it stimulates the production of chlorophyll, which is needed in the photosynthetic process. Next slide, please. And last, I want to introduce our Peroxy acetic acid disinfectant, which is called JetAg. And this is most commonly used in the field as our 5% formulation. So it's a broad spectrum fungicide, bactericide, algicide product that is safe for use on crops at the labeled rates to eradicate diseases. Its mode of action is through oxidation, and one of the most powerful oxidizing disinfectant products out there. And it's just under the ozone oxidizing capacity, or excuse me, it's just under the oxidizing capacity of ozone. And because it is a contact disinfectant product, it's gone soon after the targeted surface dries. So you don't have any concerns about residues with this product. The one thing that you do have to remember, though, is there is no residual because of the fact that it just is killing what it is contacting. The great thing about the JetAg label is that it covers lots of different uses, and it's highly compatible with other products. So you can tank mix it with many other fungicides, and that gives you that additive effect of an eradicant plus a preventative. So because it does have that wide label, you can use it in the soil, for foliar disease eradication, for irrigation cleanup, and for water treatments. Next slide, please. So that covers the biofungicides that I wanted to introduce to you. And now I want to turn it over to Steve Bogash, who is our product development and technical specialist from the Northeast. And he's going to present some data showing the benefits of adding biofungicides into your hemp crop protection program.
Thank you, Lois. It's great to be with you all this afternoon. Next slide, please, Brenda. So I'm going to present some selected research data from over the last couple of years. This one was from Oregon City. The variety is cherry wine, and this is a comparison of Botry Stop and Stargus against an untreated control for managing bud rot, also known as botrytis. You can see where the scale on the left goes up to 70%, and this was a pretty severe case in this field. We're looking at above 60% or so. Botry Stop brought it down to about 40%, and Stargus at 2 quarts to the acre got it down to 20%. So this was very, very high disease pressure during this trial. Stargus does a really nice job of managing botrytis, and as Lois was mentioning, it also helps to boost the way that plants defend themselves against disease. Next slide, please. So you can see on the right, the untreated control, that's quite a bit of bud rot and nice clean buds at Stargus at 2 quarts to the acre. We recommend a reapplication at every 7 days, and you should definitely be using a spreader sticker in the field. Next slide, please. So this is looking at Regalia for overall crop improvement. One of the things that you'll always notice about Regalia is that anytime you're spraying it in field, the crops are darker green. They're overall healthier compared to unsprayed. It only takes a couple days for that effect to take over. So these are a bunch of different rates. This is, you can see the untreated control, and this is all based on yield. And so the untreated control at just over 640 grams, then Regalia at 1 quart, getting us up near 700, and at 3 quarts to the acre, approximately 800 grams. This is reapplying this on a 7-day schedule, and you definitely don't want to be outside of 10 days. You can go out that far, but no further than that, or the ISR, SAR, primary effect of Regalia, really starts to fall off rapidly. Next slide, please. One of the things that you always see in a field where you're applying Regalia is that plants are greener. Regalia directly increases the quantity of chlorophyll A and B, and you can see here on the in the scale on the right that chlorophyll A was, and this is the measurement in nanograms per liter. The chlorophyll A is substantially increased, as is the chlorophyll B on the right. Usually this effect will take off within not that many hours, and certainly by the day after. And this is a large part of the reason that you'll see enhanced yield. The plants are simply running a whole lot better. They are running at a much higher level. And next slide, please. So this is drenched in the greenhouse. Regalia is well known to increase root mass. Definitely it makes for healthier plants. One of the slides that Govinda had shown earlier was fusarium. And when it comes to preventing root diseases, a couple drenches with Regalia prior to putting out is going to put out a lot healthier plants. I always look at the field spot, each spot in the field where you're transplanting into, that footprint has a certain value to it. So having your plants take off better, losing less plants, that's a direct economic return that you'll see. And you see this across a wide range of crops, not only larger plants, larger root systems, but a faster establishment. Next slide, please. And here you can see the difference, that plant in the middle, that dark green color, and nice full plants. And on the left, untreated, on the right, treated plants. You can just see the difference in color. This carries through. As the plants start to make buds, that's a large drain on resources. Keeping Regalia in there is going to help you to prevent powdery mildew and make for an overall healthier plant. Next slide, please. And you have to be in here ahead of time. One of the most difficult things for growers who are used to producing crops that are not organically or where you're not using biologicals is you can't wait until you see a problem. You can definitely try and intervene at that point, but being ahead of the game, you know you're going to see some of these diseases every year. You know you want to get the best out of it. So being ahead, proactive treatment, that's the best way to see a good return on these. And Lois, I believe I'm turning this back over to you at this point.
Yes, thanks, Steve. Go ahead and go to the next slide, please. So for the last section of the webinar, I want to present 2 bioinsecticides that can help hemp plants and protect them from some common pests. So Venerate is our liquid bioinsecticide with the active ingredient derived from fermenting Burkholderia rhinogenesis bacteria, which is then inactivated. So there are no live organisms in this formulation. Its fermentation media is active on foliar insects, for example, aphids and corn earworm, of which Govinda both talked about both, as well as spider, broad, and russet mites. And it's also tank mixable with many other crop input products through foliar application or fumigation. So Govinda mentioned corn earworm. I was in Southern Oregon in a hemp field last year with Rick Hilton, the entomologist here, and the applicator or the grower had applied Venerate plus BT, and it just smoked the corn earworm. So there is a lot of times there's an additive effect by putting a biological with another biological or putting a biological with a conventional. So that's always something to keep in mind. Next slide, please. So Venerate has multiple modes of action, including ingestion and feeding interference. When target pests feed on the treated foliage, it takes them about 4 to 7 days to die by ingestion. So it interferes with and it also interferes with the probing behavior of sucking and piercing insects and mites, and it causes chewing insects to stop feeding rapidly after exposure. So one of the things to keep in mind is that even though it takes a few days for the pest to die, you don't want to go out the next day and expect them to be gone. The more damage is prevented starting shortly after treatment. It has good residual control and is broad spectrum, which makes it a valuable addition to your IPM program for hemp. And also important is that it has minimal impact on benefits, which allows them to also become part of your arsenal and assist you in your pest control program. Next slide, please. The final bioinsecticide I want to introduce is Grandivo. Its active ingredients are non-viable Burkholderia subsugae bacteria and also the spent fermentation media. Like Venerate, Grandivo helps control chewing and sucking insects as well as spider mites, broad mites, and russet mites. And it has an excellent safety profile on pollinators and beneficials, probably one of the safest products that you can put out there to preserve those beneficials and pollinators. And it also has really good tank mix compatibility. Next slide, please. It has multiple modes of action. Within seconds after the target pests come in contact with Grandivo, they stop feeding and they become agitated. It also can repel them from new infestations. It is primarily a gut disruptor, and even though it takes about 4 to 7 days, like Venerate, for the treated pests to die from starvation, in the meantime, you get reduced egg laying, egg hatching, and fecundity, and so it helps you break that reproductive cycle. Next slide, please. So that covers the bioinsecticide portfolio that we wanted to present today. And so now I want to turn it back over to Steve, and he is going to present some data showing the benefits of adding bioinsecticides into your hemp program.
Thanks,
Take it away,
Lois.
Steve.
Next slide, please, Brenda. So let's look at it on, this is on 2 spotted spider mites. This is out of Oregon. And so this is eggs per 3 leaves. You can see got up to some pretty high levels here. And this is over 4 different dates, all during September, beginning to end of September, which is when you would expect them to really start piling up. Things dry out and we're at the season. You have an awful lot of pressure out there. So this is Trilogy compared to Grandivo. Grandivo rotated with Venerate and 2 different rates. And then this is compared to Preference. And you can see here that Grandivo rotated with Venerate was highly effective on managing 2 spotted spider mites. So the typical rate profile that you're going to see, this is in milliliters per liter and grams per liter. So typically the rates you're going to be looking at 1 to 3 pounds of Grandivo. Typically 2 pounds is the most common rate, and 2 to 4 quarts of Venerate. Typically we're using 2 to 3 quarts of Venerate for most purposes. Next slide, please. And here we are, adult mites, and you'll see the same kind of profile here. This mimics the research from the slide before, except this one is actually looking at adult mites per leaves. Next slide, please. As Govinda was mentioning earlier, aphids are a major problem, and you can see that Grandivo, so that's the number of aphids per number of nymphs aphids in a cage, and this is at 48 hours and 72 hours. This is a relatively moderate pressure. Oraboost was used as the surfactant here. Grandivo did a pretty good job, but Venerate at that middle rate, and that would be Venerate. I've got the note here. So that at 10 milliliters per gallon, that's actually a pretty low rate of Venerate, completely clean. That's a very low rate of Venerate being used. Next slide, please. And again, green peach aphids, this is just a slightly different study. This is looking at treatment time, and you can see the untreated control, pretty significant number of aphids. Grandivo doing a pretty good job, but Venerate just completely cleans them up. Generally, we see that Grandivo has some extra efficacy for mites, where Venerate has some extra efficacy for aphids. Here in the East, we also use Venerate for managing leafhoppers. I don't believe we have a data set that matches that today, though. Next slide, please, Brenda. And this is a study we worked on. This is Stanton Gill out of University of Maryland, looking at controlling root mealybug. Root mealybugs and root aphids are significant pests when you grow crops in media. And both of these gave near 100% control. Both Grandivo and Venerate as a drench does a great job at managing root mealies and root aphids. You do often need to reapply. Most growers, when they start having an infestation, will reapply every 7 days. Next slide, please. This is Venerate used for hemp seeds, and so we're looking at controlling a number of pests. And so yeah, this is a corn earworm study. So you'll see here the water control gave very little mortality. Venerate at 4 quarts to the acre, significant. And as was mentioned earlier, a BT like Zentari also gave significant control. Like as Lois was mentioning in the West, here in the East, we also will often use a tank mix of Venerate plus Zentari. One of the things that's really, you'll notice here, Javelin and Dipel, significantly lower levels of control. And that's because Zentari is the Aziwa strain, and I hope I pronounced that right, the Aziwa strain, and it makes a big difference which strain you are using on which caterpillar. Tank mixes between Venerate and Zentari are definitely the way to go. Next slide, please. And this is, again, a different study from Dr. Tom Kuhar, and you're seeing the same kind of thing, where Venerate and Zentari are doing the best level of control, and this is 4 days after. One of the comments I wanted to make to Govinda, because he was talking about the use of traps, and I've been working with sweet corn traps for a very long time, looking at corn earworm and corn borer, and one of the things that you regularly notice is that the traps work really well in corn, but the worms, the female moths, when they're laying eggs, will often show preference for certain crops over the traps. We certainly see that in the corn earworms. I've seen fields that are near greenhouses where they are actually avoiding the traps and eating petunias in the greenhouses. You don't see that very often, but that's not uncommon. Corn borers will always show a preference for peppers over sweet corn, especially the pheromone traps just don't seem to be able to beat certain crops. Certain crops will draw them in much better. Next slide, please. And so this just summarizes some of what we're talking about. This is, you're going after the newest hatched larva, and that's really important. By the time there was a video that was shown earlier in here in this presentation, by the time you get to those kind of large larvae where you're in final instars, these kind of products are going to have very, very minimal impact on them. You've got to be in the first, second, maybe even into the third instar, and that means you are scouting constantly. Using the sweet corn type traps where you're using the pheromone traps is probably one good way to go. But, you know, once you start to get buds forming, being proactive, I know it's not a very IPM practice, but being proactive with a tank mix of Venerate plus Zentari is probably a good way to go, because it's very, very difficult to spot first instar caterpillars. They're barely the size of a thick pencil lead, and you just don't want to wait to the point till you have large adults. We just don't have the tool for managing them at that point. And it takes a lot of water. Often when we see failure to control, it's a lack of water. You're just not going slow enough and putting enough water on to really push that product down into a dense canopy. Next, please. And again, you've got to be proactive. Biologicals work best when you're keeping things under control, not when they're getting out of hand. And somebody was asking about why this is so important compared to synthetics, and a lot of it is you're trying to prevent overlapping generations. Most of the biologicals are not very good on eggs, and so you've got to get the nymphs or the young adults. And if you let them get older, often they are resistant to it, and the eggs are largely resistant to it. So the idea is to get in there early, keep them under control so you prevent overlapping populations. Next slide, please. And I think I'm handing this back off to Lois or Brenda here.
Thank you, Steve. I believe Lois added this in. We do have a Hemp IPM guide. You can go ahead and find this on our website as well if you'd like to download it and print it for yourself. We do have a national sales team. If you'd like to take a quick picture or just take a screenshot of this for future reference, we do have all of their contact information up above. And we also have our technical support team. We have our PD and team all around. So if you'd like to keep this for future reference, feel free to do so. Okay, now we can take a few minutes or we'll stay here for the remainder of the time. If you have any questions for any of our speakers, I believe Steve and Lois are still on, as well as Govinda. So if anyone has any questions, feel free to ask them.
Brenda, I think we had a question for Govinda. Sibi wants to know if beneficial plants such as alyssum will aid in keeping pests at economic threshold. Govinda, can you address that question, please?
Can you repeat the question again? Can you read one more time?
Sibi wants to know if beneficial plants such as alyssum will aid in keeping pests at an economic threshold. In other words, can you use plants that will help build up your predators and your and that to help you with your pest control program?
That's a great question. Actually, we don't know exactly how it will work on the hemp, but for example, in the lettuce, to control the lettuce aphid, which is a major pest in California, in Arizona, they have sort of like the different flowering plants they put into the border or kind of like between the plants, and that helps to attract the syrphid fly, the predator, and that really control the aphids. Particularly this we call as a conservation biological control. You have a kind of making the environment that attract the natural enemy and then help to suppress the pest populations. And we don't know exactly in hemp how it will work, but definitely in the future that also is a good research question, that whether we can have sort of like flowering plants and that attract the trichogramma as an example, and then control the corneogram. So we don't know exactly, but that's a good question to us. Is there any question? Go ahead.
Steve, can you address the question from Saul? He wants to know if the venerate, he says the venerate label claims to interrupt molting. Wouldn't that hurt larval stages of beneficials? And if not, please elaborate.
So it's interesting, the mode of entry and mode of action of venerate. The primary mode of entry, this is the reason that both venerate and Grandivo are so safe for beneficials, is the primary mode of entry is through ingestion. So the insects have to eat it. And this is relatively, for us as a research team, this is relatively new news. We always wondered why it was so soft on beneficials. So it's got to be ingested, and then it is a biochemical that is a stomach poison. Both Grandivo and venerate have different pathways, but they're both stomach poisons. So in order for what we're trying to control are insects that eat our crops in various ways. And so They're the ones who are going to consume it. Beneficials are simply not going to consume it. You can spray it on them and it does not have any impact. It's got to be ingested. That is the primary way that they work. And so an insect that ingests venerate is obviously going to be in distress and is going to have problem molting. It just simply interferes with so many things that are happening. And this can happen very quickly. Actually, it stops them from eating. It stops them from eating more quickly. So they are a combination of starving to death and being poisoned at the same time. But since beneficials are not eating it, they are not going to be affected by this.
Thank you, Steve. We do have a few more questions. Have there been tests conducted to quantify the microbial level as a result of biological use?
Steve, do you?
So I guess that one's fallen back to me
Yeah.
too. Okay, so Regalia, that's obviously not a problem. It is a plant extractor. It's not a concern. Stargus is a colonizer, much like a lot of the natural, like Botry Stop is. It's a colonizer, and so it will affect the final bacteria load, especially if applied close to harvest in the last couple weeks. So you will definitely have an impact on the overall microbial level. For growers of non-hemp cannabis, we recommend at least 3 weeks before harvest, for that final application, just like you'll find with a lot of the other biological products in there. As far as Grandivo and Venerate, both of those are non-living. They've been fermented, and they are dead before you get them. And so it's the products that the bacteria make that are actually doing the primary level of control, not living bacteria out on the leaves or on the buds.
Thank you,
Good
Steve.
question.
Thank you. We do have a couple more questions. Enrique asked, is TerraGrow available in Arizona?
TerraGrow. That's not one of our products. I don't think we mentioned TerraGrow. And so I would check with the folks at, that is by the BioSafe product. I would check with them.
Perfect, thank you. And one more question from Saul. So pirate bugs that drink from plant are subject to death from venerate?
Not according to our research. Aureus, the minute pirate bug, it eats other bugs. Therefore, it is not going to be consuming enough product to make a difference.
Thank
And
you.
I think I saw one question. Somebody was asking, it sounded like it was John Jemison from Maine was asking about a respirator for Stargus. That was one of the earlier questions I saw on there. And the simple thing is that you are required, according to the label, you are required to use a respirator when mixing, but not as an applicator. I just looked at the label a few minutes ago. I didn't memorize that.
Thank you, Steve. We have a question from Benjamin. Are there any problems with using Regalia for someone who is processing all of their flour into oil? Any evidence of Regalia affecting the end oil product?
None that I've ever seen, and that's a great question. What kind of residues you get? So these, all these products, as Lois mentioned earlier, they're MRL exempt, and so they are exempt from all residue levels. We've done some research in wine. I know it's not the same crop, but we've done some research in wine looking at phenolics and other residues. It doesn't have a significant impact there, but because it does turn up the ISR, SAR biochemical pathways, I am sure it is having some impact. Whether you can detect it in the oil or not, I do not know.
Also, can I add to that, Steve? In talking to our cannabis specialist on staff, I've been told as the salesperson that to tell our customers that by the time you get to about second week of flower, Regalia has really done its job. And so if you feel like you still need a fungicide, you'd be better off to switch to Stargus. That Regalia, because it takes time for the plant signaling and everything, that you don't really need to use it after about the second week of flowering. And so that also helps. Not that it would never test out, because there's nothing in it that would test out, but you wouldn't have to worry about it if you stop like second week of flowering and then switch to like the live bacillus if you feel like you need some botrytis control to get you on into harvest.
Thank you, Louise. Go ahead.
I was just going to say, somebody asked if said they were having troubles with the California, getting their information in for California, so they put their license in. We'll make sure that whatever state you have, that you will get your credits. And if you have any question about that, be sure and respond back to the email you get from us.
And I was looking in the chat. I saw a couple more chats come through since we've got a little bit of time. Here in the East, we have significant leaf spot problems. And so we see things that are septoria related, frog eye related. There's a number of different leaf spots that'll get on. It doesn't have a huge impact, but you can certainly get some major defoliation. Stargus is a much better product for managing leaf spots. It's very effective. We use it on quite a few crops, and it is a good practice when you're dealing with leaf spots to tank mix Regalia and Stargus. They are very friendly with each other.
I think, Steve, there's a question from Saul about where can we find the research on effects on aureus?
So I would contact me. Send me an email. Or if you know whoever your rep is, your PD rep, I couldn't tell where that person was located. I cover Virginia and North and the Southeast and Ohio and East, but we've got other PD folks probably closer to you, and so you can grab our emails off of the website. I saw that it flashed up for a moment, but I can be found at sbogash@maronebio.com, and I'll either forward it to whoever your person is. In-house research, most of it is university research or private. And so asking about our research, this is one of the places it's really great to work for a company like Marone. The overwhelming vast majority of our research is done outside. Private researchers and universities, most of the research that we do in-house is developmental. By the time we get out in the field, we are looking to see how things actually work in the real world or in small plot work.
Thank you, Steve. I saw a question regarding the test questions for California credits. I can go ahead and put up our next slide, which includes a QR code. You can go ahead and scan that to take the quiz to receive credits for California, and you will be receiving your certificate within 1 week.
I saw another question, same question regarding where is it all published. Some of the trials do end up in journals, like Stanton Gill's work on root aphids. That he had published that just recently. Much of that work is done under contract for us, and unless the specific researcher has elected to try and submit that to a journal, they're just research reports that we have. Aureus Trial University, that's good. But I'll gladly get you whatever research we have that we can share with you. Most of it's public knowledge. And Saul, just email me.
Somebody asked too if they have to take the test if they're an Oregon attendee. We usually ask everybody to take the test just because that assures both Oregon and Washington that our attendees were engaged all the way through to the end. So yes, please do that. It helps me when I'm submitting those credits. Hey, did we get them all?
I think we are wrapped up.
Somebody said they're using the camera function to scan this with no luck. You are going to send that information out, right, Brenda, so that they can access it after the webinar.
Yes, you're able to access it later. You can also email myself. I would put it in the chat. There's also a link in the chat if that works better for you to take that quiz.
If anybody has more questions too, you're always, our website shows the contact information for all of the staff members all over the United States. And you can always get a hold of whoever is closest to your area to be more specific. And then Govinda has been great. He's just so helpful. He's a great resource no matter where you're at too. So.
Thank you, Jess.
I believe we are okay with questions. We will be staying around for a few minutes, but I would just like to thank you all for attending today, as well as our speakers. Thank you so much for taking the time to join our webinar today.
Brenda, I'm going to go ahead and sign off if
Sounds good, Lois. Thank you so
Okay,
much for
great.
joining us.
Thank you.
Thank you.
Thank you for-
Bye-bye.
Mhm.